Auzubillahiminashaitanirajim. Bismillahirrahmanirrahim. In the name of Allah, the most merciful and beneficent. My presentation topic is fermentation industry, presented to Dr. Hammad Majeed, presented by Khadija Tariq, role number 2, and subject is industrial chemistry. The table of contents are fermentation definition. At first, I will explain what is fermentation, then its discovery, its objective, its performance, types of fermentation. We will be now about the types of fermentation, but we mostly know about two types of fermentation: alcoholic and lactic acid fermentation. But I will be explain other types of fermentation with them. Next one is fermenter or bioreactor. Fermenter, I will be explain what is fermenter or bioreactor. Fermentation process steps, those steps which are performed in industrial scale, I will be explained about it. In next industrial scale diagram, I will be explain the diagram about to where fermentation takes place, and then factor affecting the fermentation: pH, temperature, what type of factors affecting fermentation. Next I will be explain operating system, bioreactor types. At last I will be explain products of fermentation. Many types of products are formed in fermentation by using fermentation. Only we not form the ATP, we form our other products and we use it in daily life. So fermentation is very important in our daily life. At last I will be explain about the reference in which I take this type of data and oth- other things. The first one is fermentation definition. What is fermentation? We will be know what is fermentation. Um, at first, I think when I did not learn about it, did not take the material to the internet or other websites, books. So, at first, I think fermentation is a process in which we break down and molecules of sugar into small molecules, simpler molecules. Then the ATP is formed in energy is released in the form of ATP. ATP means adenosine triphosphate. Then it is a living cell. I know that it performed only by microorganisms, bacteria. Now, but when I take these information, then I know not only bacterial cell or microorganism cell perform this process. Mammalian cell, animal cell, uh, other cells are also used to perform this process. So now I read the slides. It is the process by which the living cell is able to obtain energy through the breakdown of glucose and other simple sugar molecules without requiring oxygen. It means that it is an aerobic process. Fermentation results in the production of energy in the form of two ATP molecules. I will be discussed about it, and produces less energy than aerobic process of cellular respiration. Derived from the Latin verb fervere, meaning to boil. It is derived from the Latin verb and that means to boil. Now we move in next slide. That is discovery of fermentation. At first, Louis Pasteur is a scientist who discovered fermentation. He discovered the type of fermentation, that is alcoholic fermentation or ethanolic fermentation. So we can say that alcoholic fermentation is the first discovery of fermentation. Louis Pasteur in 19th century used the term fermentation in a narrow sense to describe the changes brought about by yeast and microorganisms growing in the absence of air anaerobically. He also recognized that ethyl alcohol, carbon dioxide are not only the products of fermentation. That means lactic acid is also formed in fermentation, and other things that are butyric acid, Propionic acid, diacetyl, other products are also be formed in this fermentation. Louis Pasteur at first recognized it, but he does not discover these products. He only discovered ethyl alcohol and carbon dioxide. Now we move the objective of industrial chemistry. What is the objective of industrial chemistry? The main objective of industrial chemistry is to produce highest quality and quantity of a particular product. That means we need a desired product which have highly pure and can formed in large amount by combining the works of different disciplines such as that microbiology, biochemistry, genetics, chemical and bioprocess engineering, mathematics, and computer science. We can say that fermentation is not in present in bio or chemistry. Fermentation is present in chemical process. In fermentation, mathematics and computer science is also be present. By combining these six disciplines or these six topics, these six subjects, we perform fermentation process. And by using fermentation process, we produce now made by industrial fermentation with generally modified microbes such as lipase and invertase. In certain cases, production of biomass itself is the objective. Example, baker's yeast and lactic acid bacteria starter cultures for milk product synthesis. I will be explain these two things. That is at first objective. We uses some enzymes which are very important. That is lipase, which break down of lipid, invertase, which breakdown of the urea into the zymase or other products. So I can say that by using fermentation, we form mostly important enzymes. In certain cases, production of biomass means that we form the milk product synthesis, milk products by using fermentation. In next slide, I will be discuss about performance of fermentation. What is the performance of fermentation? Fermentation is performed in small scale in also large scale. In small scale, it is performed in very previously or in very previous years. Now, fermentation can be performed on a large scale, often as several thousand liters in volume in many industries like microbiology, food, pharmaceutical, biotechnology, and chemical. These these five industries, fermentation process can be takes place and the products can be formed. Next is types of fermentation. There are six types of fermentation. Other types are also present, but I will be discussed about six types of fermentation. I know mostly these types of fermentations are also are used. Um, Lactic acid and alcoholic fermentation is widely used. Other fermentations also takes place in large scale. There are two types of lactic acid fermentation. Number one is lactic acid Homo fermentative. Number two is lactic acid heterofermentative. I will be explain a difference between them in the following slides. Number three is propionic acid fermentation. Number four, diacetyl and 2,3-butylene glycol fermentation. Number fifth, alcoholic fermentation. Number six, butyric acid fermentation. In these fermentations that are the fermentation in these fermentation, the products that are formed in that are formed are are present in their definition or in their headings. For example, in butyric acid fermentation, the product that is formed in this fermentation is butyric acid. In alcoholic fermentation, the product is alcohol. In propionic acid fermentation, the product is propionic acid, and lactic acid fermentation, the product is lactate or lactic acid. Types of lactic acid fermentation. There are two types of lactic acid fermentation. Number one is homofermentative, number two is heterofermentative. Homofermentative is decomposition of hexoses by glycolysis, single fermentation product lactase, uh, energy generation 2 mole ATP per mole hexose. Heterofermentative decomposition of hexoses by phosphoketolase way fermentation products carbon dioxide, ethanol, lactate. Energy generation 1 mole ATP per mole hexose. In homofermentative, we can say that it is better than hetero because by using it, if we need energy, then homofermentative is the best. If we even need the products that are formed that are carbon dioxide or ethanol, then hetero is good. A homo means same. It means only lactic acid is formed, that is same type. Hetero means different. That means not only product lactate is formed, but other products are also be formed that are called side products. In homofermentative, glycolysis can be performed, but in heterofermentative, phosphoketolase process can be performed. The ATP molecules are high in number in homo and low in number in hetero. What are the pathways of homofermentative and heterofermentative? As we say, the circles are present in this slide: glucose and lactose, glucose, carbon dioxide, ethanol, lactate. At first, glucose is converted into 1,6-bisphosphofructose. Two ATP molecules are converted into ADP molecules. We can say that two ATP molecules are reduced in it. Then, by the conversion of 1,6-bisphosphofructose into 2-glyceraldehyde 3-phosphate, and then 2-glyceraldehyde 3-phosphate is converted into 2-pyruvate, and then 2-pyruvate is converted into 2-lactate. When 2-glyceraldehyde 3-phosphate is converted into 2-pyruvate, 2-NAD is converted into 2-NADH. That means it is a dehydration process step, not a process. It is a step, dehydration process step, and 4 ATP molecules are formed. At previous slide, I will be explained that two ATP molecules are formed in lactic acid homofermentative. That is, two ATP molecules are reduced, 4 ATP molecules are formed, so we minus 2 ATP molecules are formed in the homofermentative. Now, glucose is converted into 6-phosphogluconate, then is converted into pentase phosphate, then is converted into glyceraldehyde 3-phosphate, then converted into pyruvate, at last convert into lactate. Uh, these side products are also formed. We see it pentose phosphate in third step is converted into acetyl coenzyme A by the addition of coenzyme A. Then removal of coenzyme A, acetaldehyde is formed. Then acetaldehyde is converted into ethanol. One ATP molecule is formed in first step, not is formed, is used. Two ATP molecules are formed in the 1, 2, 3, 4 step. That means at first step, one ATP molecule is used, and at fourth step, two ATP molecules are formed. Two formed, one is used. So we subtract 2 with 1, and one ATP molecule is formed in heterofermentative lactic acid fermentation. And other processes also takes place, that is dehydration, decarboxylation in heterofermentative. But in homofermentative, only dehydration takes place. We can say that coenzyme is only heterofermentative. It is not used in homofermentative fermentation. Now we move next type, that is propionic acid fermentation. At first we can say that lactate is converted into pyruvate. Those lactates that are formed in the lactic acid product are converted into pyruvate. Pyruvate are converted in two things, oxalate or acetate. Now oxalate is converted into malate, malate is converted into fumarate, fumarate is converted into succinate, succinate converted to 2-succinyl coenzyme A, 2-succinyl coenzyme A is converted into methyl malonyl coenzyme A. 2-propanyl coenzyme A, then 2-propanoate